All right, we're going to go ahead and get started. Hello, everybody, and welcome. Good morning, good afternoon, depending on where you're located. My name is Samantha Jameson. I am the Senior Marketing Specialist here at NBS Scientific. Today, I am joined by Cathy Roesch from Basque Engineering Science, and we are here to present a webinar for you on frozen sample aliquotting technology and the CXT 353. So a couple announcements before we get started. Today's webinar is being recorded, so if you have to leave early for any reason, no problem. We will share a recording of the webinar with you before the end of this week. Your microphones and cameras are automatically muted during the webinar, so if you have any questions as Cathy is presenting, I encourage you to put those into the Q&A box on the right side of your screen. There will be a time towards the end of the presentation, actually after the live demo, where we will ask Cathy our questions, whatever they may be. So I encourage you to put those into the Q&A box as we move along with the presentation today. If you would prefer to email us personally or privately your questions, that's totally fine as well. You can send your inquiries to the email address that's on the screen. That would be info@nbsscientific.com. And if you are located outside of the US, that's totally fine. We'll make sure that your inquiry gets passed along to the appropriate sales team. So before I pass things off to Cathy, I just want to share a little bit more about NBS Scientific and who we are and what we do. So NBS Scientific is a distributor of laboratory products for the life science industry. We are part of an international distribution network, so we have sister companies located in the following countries: in Belgium, Denmark, France, Germany, Spain, Switzerland, the Netherlands, the UK, and of course the US. Our universal portfolio includes things like sample collection labware, sample stability reagents, your sample storage tubes and racks and caps, and then everything else from barcode readers, tube labeling systems, cappers, decappers, laser markers, rack thawing stations, tube handlers, and then of course what we are discussing today, which is the frozen sample aliquotters. So I know I just listed off a whole bunch of products. So if anything happened to pique your interest just a little bit, I encourage you to visit our websites for more information, and then of course reach out to us via our chat function on the website or send us an email with any questions that you might have. So then without further ado, I'm going to pass things over to Cathy, and she will begin by talking a little bit about frozen sample aliquotting and why it's important to laboratory science. So Cathy, when you're ready, feel free to take the floor.
Thank you, Sammy. Welcome, everybody. Thank you for joining us today to learn a little bit more about the frozen sample aliquotting technology. And we're going to do a live demo today of the CXT 353. So when we talk about frozen aliquotting, my favorite saying is because thawing is not cool. Next slide. All right, so NBS and Basque Engineering have partnered up. We're partners in frozen sample solutions for stable, reliable, and reproducible results. Next slide. Why is frozen sample aliquotting important for the laboratory science? Well, there's 7 things that I like to discuss. The first one is that it improves sample integrity and preservation. The second thing is it eliminates sample contamination. The third thing is that it increases sample utilization and reaccess for later on. You work with the samples safely. It delivers sample consistency. It ensures reliable and reproducible data. And last but not least, it is time efficient for the scientists. Next slide. All right, so we're going to go over number 1, which I talked about, which is improved sample integrity and preservation. So how does it do that? It does that by eliminating the freeze-thaw cycle that can degrade analytes, causing bias data in downstream analysis. The other thing that it does, it stabilizes labile molecules. Next slide. All right, the second thing, eliminates sample contamination. So our patented technology uses nuclease-free coin probes to extract out the aliquots pores from frozen biosample, depositing them into cryotubes free of contamination. Both the parent and aliquot samples are maintained at a temperature of -80 or colder throughout the entire process, ensuring preserved, high-quality samples for use in downstream assets. Next slide. All right, number 3, increases sample utilization and reaccessibility. One or more specific size cores are extracted out, leaving behind an intact parent sample. The parent sample is then returned to the -80 freezer with the ability to reaccess that later on free of contamination. Next slide. Working with samples safely. So it eliminates manual slicing and cutting of tissue. It eliminates handling of raw feces, and it cuts down on aerosol exposure. Next slide. All right, delivers sample consistency. Generates and distributes small, uniform, volume-specific aliquots in a controlled and efficient manner. The coring technique is always from the top of the sample all the way down to the bottom of the sample. Next slide. Ensures reliable and reproducible data. Aliquots thaw faster than a full sample, improving sample consistency and reproducibility of data and results. Samples kept frozen retain original analyte value because they're not degraded due to the freeze-thaw cycle. Therefore, reaccessing of that same sample later on will produce the same original results. Next slide. Savings for time. Saves time for scientists. Time savings improves workflow with a 60-second coring cycle versus sample thawing, aliquotting, and refreezing. Next slide. All right, so now we're going to go into the frozen aliquotting process. Next slide. All right, so how does it work? So I hope everybody can see the instrument. All right, so the instrument is cooled down using liquid nitrogen. And as you can see in my slide, there's a little porthole that you put the LN2 in. It reaches -80 in 10 minutes. Samples are then removed from the -80 freezer and placed on the instrument. In this model, the 353, we have one sample fixture. So you're going to core one sample at a time. Samples are then removed from the freezer, put on the instrument. The coring probes are then placed on the instrument and chilled to -80 at the same time. Next slide. Sample is then placed in the coring fixture. Coring probe is inserted into the coring head. Sample is then cored using specific depths set by the end user within saved protocols. Next slide. The frozen aliquot is extracted from the frozen parent sample. The frozen aliquot is deposited into another cryo tube sitting within the destination carousel that's also at -80. The aliquot remains frozen during the entire coring process. Next slide. Coring probe is then removed from the coring head and is replaced with a new probe. The next sample or the next aliquot, same sample if you choose, is cored, and then the process is repeated. So what I need to say at this point is that from the time that you insert the probe, you core your sample and it's deposited, it is a 60-second cycle. Next slide. Yes. All right. What kind of biospecimens can I use with the frozen aliquotting? Next slide. So we've done feces, plasma, buffy coat, urine, tissue of all kinds, heart, placenta, muscle, liver, pancreas, whole blood, serum, brain, saliva, and a multitude of buffers. Next. What types of research can benefit from the frozen aliquotting? So right now in the United States and in Europe, we have people that have our instruments that are performing microbiome studies on both human and animals, cytokine studies, DNA, RNA studies, tissue pathology, both in human and animal, antibodies and PBMCs. We're still working on that. That's a new application and it's going well. COVID studies, mitochondrial studies, small and large molecules. Biobanks are using it for QA, QC, and buffer studies for drug manufacturing. Next slide. What kind of research labs are using the CXT 353 for frozen sample aliquotting? So we have people that are in the pharmaceutical arena, hospitals and universities, animal research facilities, biobanks, biomedical research organization, and we're coming up on people that are now using it in food and nutrition. Next slide. All right, so now we're going to do a live demo. So I'm going to move my computer so that you guys can see. I'm going to get up. So the first thing we're going to do is we're going to fill the LN2 tank with liquid nitrogen. I'm going to put on my special cryo gloves. I've already preset my probes that are now sitting in the probe nest, because what we want to do is get the probes and the instrument cooled down to -80 all at the same time. Once it's at -80, then you want to take your sample out of the -80 freezer. So let's go. And what I'm going to do is slowly fill up the LN2 tank. If you do not have an LN2 tank in your lab, because some of our customers do not, it can also be filled up with a dewar. On the screen, we have in place the fill volume for the tank, which you monitor, and the temperature. I have filled up my tank. I went from room temperature, and I'm already at minus 13 degrees. So we're going to wait until it's at minus 80. It takes about, like I said in the slides, about 10 minutes or so. It will depend on your lab. The warmer the lab and the higher the humidity, it may take a little bit longer, but the colder your lab, it'll go down even faster. So I'm already at minus 20, and I think we're at 3 minutes. So what I should recommend, or what I should tell most people, is that you can put, once you put your instrument to minus 80, we do have a sample section so that you can actually take out a couple of samples out of your minus 80 freezer and keep them at minus 80 on your instrument, so that you don't have to be going back and forth to your freezer. We do have a spot on the instrument that will actually hold your samples. So for today, I already put a sample in the middle of the fixture just to save time. So we're already at minus 36 and keep going down. So Sammy, is there a way that if somebody, until we get to minus 80, if there are any questions, is there a way that we can?
Yeah, so there's the Q&A box on the right side of your screen. So at this point, if you have any questions for Cathy, please put your questions in there, and we can go through a couple maybe as we wait. Yeah.
Right, we're at minus 50. So as you can see, it's very easy to fill up the tank. Doesn't take very long. It is a 5-liter tank. Trying to think of what, it's so hard to remember everything to tell you. We have 3 different size probes. And with the different size probes, that will determine the size aliquot that you can program into the instrument to remove from your sample. We make the sample adapters to fit any tube. So whether it's a 50 ml conical tube, a 2 ml cryovial, a 1 ml, it doesn't matter if it's a Nalgene, a Fluex, a Micronic. We can make any adapter to fit any tube. The instrument will hold up to 20 protocols. So if you're doing multiple studies on multiple types of biofluids, you can program your different protocols when you're working with each different specimen. That way you're not changing back and forth the volumes. All you got to do is change your protocols. All right, we're at minus 80. I think that was 8 minutes, so we're ready to core. So the way that it works is that you're going to take the probe that's already pre-chilled. You're going to insert it into the coring head, and you're going to put the safety guard down. The safety guard serves 2 purposes. One, it protects the end user, and number 2, it protects the actual sample that is sitting in the deck. All right, so then you go forward. What I like about this instrument is that it has a laser, and you can pinpoint exactly where you're going to core in the sample using the laser. And we always tell people you want to core from the top of the sample all the way to the bottom. That way you get a great representation. Forward. So I've spotted with the laser where in my sample I'm going to go. Now it's going to core into my sample. You're going to lift up the guard. You're going to pull it over to the destination sample where you're going to then deposit the core. It's going to go down. And then you remove the probe, you throw it away, you take your tube, and for this, oops, here's my frozen core. Everybody can see that. You return it, and then you continue on. You put in a fresh new probe. You can now put in the next sample, or if you want to take a second core from the same sample, you can do that. And that's why there's no contamination, because you're using the core once. You're going into the sample with the probe, you're extracting out a core, you're depositing it, you're removing the core and probe, you're throwing it away, and you're starting again. So that eliminates any contamination. Now, with the sample, if I were to remove my sample, which in this case, it's just clay, I've got one little hole, but this now is still frozen. It's not thawing. I can put the top on this and then put it back in the freezer. And now I have re-access of this same sample for later on. And that's it. It's very simple, very easy, and very fun. Thank you. Next slide.
We do have a question, actually.
Sure.
So we got a question that asked, can the instrument hold a rack of 96 tubes or a biorad plate of 96 samples? And then can it perform dilutions with a buffer?
So the answer to that question is not in this model. If you are doing 96 well plates, you would have to go to our next model up, which is the 425. The 425 can actually core from 96 well plates to 96 well plates, or from tubes to 96 well plates, or from 96 well plates to tubes. That model will not do dilutions. If you want dilutions on top of that, then you have to go to our CXT 450, which is the next model up. And that model will actually do all of that and will do the dilutions. However, depending upon, it's not like a Hamilton where it's a head of 8. It only does one, it's got one pipetter with one head, so it does it one at a time.
Okay, great. A follow-up question for that is, what is the smallest amount that it can actually core from?
Great question. So right now, the smallest tube that we've been able to core from is probably a 0.5 ml centrifuge tube. We can core from the little tiny fluidics tubes. I think they're also 0.5. And then with the 3 different size probes, we have a baby probe that will take anywhere from 3 microliters or 3 megs up to about 30. The next size up will take anywhere from 10 microliters up to about 150 microliters. And then the next size probe takes anywhere from 50 microliters up to 300. And then we have custom probes. So if you need, we have a customer that does animal tissue and feces, and they need 1200 mgs per core. So we have designed a 8mm probe for them, and they use that. So if you need a specific volume, then we can create a specific size probe for your application and for your samples. Did I answer that?
I think a second follow-up to that, they're asking, maybe this is for the 425 or 450, but
Yeah.
what is the smallest aliquot volume and what is the largest aliquot volume, and can it do varying dilutions?
So depending upon your sample size, once again, it can take anywhere from 3 mgs or 3 microliters all the way up to whatever you need, because that instrument is flexible enough. It holds the same probes the same way. You program in the same types of protocols. So depending upon, let's say you have to take 1,000 mgs. In order to take 1,000 mgs, your sample size has, or your tube has to be fairly large, but you're going to have to take multiple cores because I don't, with a 50 ml, and even with the urine from top to bottom, the max you're not going to get 1,000 in one. That's too large, but you'll have to take multiple cores. As far as the multiple dilutions, currently that is not on the 450, but if that's something that you need where it takes multiple dilutions like on a Hamilton, we can discuss that offline, and that would be a custom instrument.
Great. We'll give everyone another minute or two to put their questions in the Q&A box. So I encourage you to keep asking away. This is we've moved into the Q&A session now, so please keep asking your questions. They've been really great. I guess while we wait for further questions, Cathy, do you want to talk about, like if someone were to purchase a CXT, what does the maintenance look like for it? And yeah, can you talk more about that?
Sure. So that's the other thing that I like about this instrument, being a med tech and being a scientist and working with all different types of instruments. This instrument is so easy to maintain. So basically what we tell people is all of the parts that are on the deck are removable. So at the end of the day, when you're done using it and the instrument is thawed out, you remove everything, you put it on a paper towel overnight to air dry out. Usually we tell people, maybe depending on how often you use it, we tell everybody maybe every other week you take all the different removable parts and you rinse them with maybe 5% SDS solution. Then you do a little bit of a rinse, and then I'd say to everyone, just take your 99% ethanol, spray it on it, and let it over the weekend dry off. That's it. That's all you have to do. There's nothing more. And then what we do is we do recommend a service maintenance contract, which is a PM that we come in once a year. We clean the instrument really, really well. We take it apart. We look for, if the motor needs to be greased. We look to make sure that everything is clean and nothing has been, worn. So we do recommend that, but as far as like a daily, a weekly, a monthly, there's nothing that the end user has to do other than to maintain your nest. And that's it. It's a piece of cake.
Awesome. Thank you. Another follow-up from, I think this might go along the lines of a more custom instrument, but would it be possible to have a head that could aliquot with a multiple pipette head like the Hamilton or another liquid handler?
For the liquid handler part, yes, that would be a custom instrument. If you're looking for a multiple head that can core into the samples so that it's coring 4 to 6 samples at a time or eight samples at a time. No, we cannot do that, unfortunately. However, with the 450, you can program into the computer and you can actually put multiple samples on the instrument that will stay frozen at the -80, but it will still only core 1 sample at a time to 1 core. Then, so for instance, we do have customer, I guess this might make more sense if I kind of give a real life situation. So we do have a customer that is in a pharmaceutical company. They are doing microbiome. So what they do is they actually take 6 cores from each sample and they deposit 3 into a 96 well plate and 3 into a tube. And then the different departments take those aliquots and do what they do. But before they do that, they will run a run where they will aliquot into the tubes and the 96 well plate, their frozen aliquots. Then what they'll do is they'll switch the plates and they'll put their reagent on the instrument, and then the pipetter can come over and the pipetter then puts the reagent or the buffer into the wells or the tubes. And then from there, they go ahead and they run their assay. If you're looking for, at that point, a multiple head that will then, like the Hamilton, that will go into the reagent, come over, and then add the reagent into the microwell plates or add them into the tubes, yes, we can do that. That is a custom instrument. So that we can do. That customer didn't want to do that. That customer wanted to do 1 at a time. They had their reasons, and that's fine. The whole point of custom instruments is that we're trying to make it work for you specifically, for your application and what your needs are.
Awesome. Don't have any other questions at this time, but Cathy, is there anything else that you want to say about frozen aliquotting or the CXT?
So I've been doing this for 10 years. I love this instrument. I mean, we are worldwide. We have instruments all over the country. We are doing some exciting new things coming up with some of our potential customers, which is really cool. I've seen this instrument grow from a lower base model, which was a lot slower, which we no longer sell. That was a 350, to the 353, to the 425. And the 425 was really born from a pharmaceutical company looking for a custom instrument that what they do is they have their buffers in different concentrations frozen. And what they wanted to do was core into the buffers, put them into the 96 well plate, add their drugs, and then run their assays. So that's how the 425 was born. And that's really interesting. And that's cool because from there, then we went to another pharmaceutical company, which was the application that I just talked about, the microbiome, where they're doing studies on diabetes and IBS. And then that's how the 450 got born. So for me to see all the different applications come along to create bigger and better frozen aliquotting to meet the research needs is, to me, exciting. I love what I do. I'm very happy that, you know, this instrument is doing very, very well. The technology is getting more and more useful for other things. We have a potential customer for cord blood. So to me, it's exciting, and I'm hoping that you guys find it exciting too. Feel free to contact us anytime if you want to learn more. If you want to do a private demo, we can do a more one-on-one demo. I love to teach, and I hope you guys love to learn.
Awesome. Yeah, it's definitely exciting to hear about the different ways in which people are using the instrument and the different research that they're doing. We did have another question come through that asked if the machine could work with a LIMS database or Excel spreadsheets.
Not for this model. So the 353 totally independent of that. So is the 425. If you are looking for something where you generate where you can use a barcode reader, so that's what the 450 can do. We can add a barcode reader to that, which will then read the barcode, generate through Excel a work list, if that's what you're asking. Can it then take the results off of the instrument and feed it automatically into the computer? The answer is no, not now. That's a little bit more of a sophisticated, interface. It's more that we can generate through the barcode reader a work list, and then you would have to use that work list to run your assays and then manually enter the data. I wish I could do it all.
All right, those are all the questions that we have right now. So this just takes us to the last slide, which just has some contact details. So if you would like to discuss, your specific research application, or if you are interested in a custom instrument, or just any questions in general, please feel free to contact us. You can chat with us online. You can send us an email with your questions to info@nbsscientific.com. If you're located in the United States, you can also give us a call as well. As I mentioned at the beginning, if you are located outside of the United States, still send your, I encourage you to still send your inquiries to the email address on your screen, and we will absolutely connect you to the appropriate team so that we can get your questions answered. But yeah, that brings us to the end of our webinar today. I want to thank everyone so much for attending, and Cathy, as always, for talking about frozen aliquotting and sharing all of her knowledge about the technology, and also doing a live demo of the CXT 353. So thank you, Cathy, for your time. And yeah, I encourage everyone to contact us and find more information online. But thank you so much for your attention, and hope you have a great rest of your day. Thank you so much.